Welcome to this explainer, where we're going to turn you into a full-blown plant detective, totally ready to solve those weird medical mysteries popping up in your garden. I mean, you know the feeling, right? You walk out to the backyard, take one look at your favorite plant, and just realize something has gone horribly wrong.
And that brings us to the ultimate crime scene question. Why did your plant just die? Was it just some environmental stress, maybe a sudden drop in temperature? Or is there an invisible, living culprit at work? Well, today, we're going to learn exactly how to figure that out.
So let's open up the case file and lay out our docket for today. We'll be looking at the Plant Detective's Toolkit, reading leaf patterns, deciphering diagnostic terminology, figuring out if it's fungi, bacteria, or a virus, and finally doing a deep dive focus on the Alfalfa Mosaic virus.
All right, moving right into Section 1. The Plant Detective's Toolkit are foundational tools.
So let's jump right into the disease circle. This is really our first major clue. We've got to differentiate between biotic, meaning living attackers, and abiotic, which are non-living stressors. Biotic factors, those are your fungi, bacteria, viruses, and insects, the things actually infecting or feeding on your plant. Abiotic factors are environmental. Think drought, poor drainage, bad nutrients, or even chemical injury. And here's a pro tip. Before we jump to the conclusion that a disease is contagious, we always have to rule out those abiotic stressors first. Why? Because honestly, they're usually the easiest things to fix.
Now, one absolutely powerful concept in plant pathology is the disease triangle. You can think of it like this heavy-duty vault door that needs 3 very specific keys to unlock an infection. You need a susceptible host, a favorable environment like, say, super high humidity or soggy soil, and of course, the actual pathogen has to be present. Here's the beauty of it. If you remove just one of those keys, maybe by changing how you water to alter that environment, the disease literally cannot take hold. It's a total game changer.
Okay, so to build out our case file, we follow a pretty strict procedural checklist. First up, host identification. You absolutely have to know what plant you're dealing with and what its normal needs are. Second is the problem description, like what exactly is changing here. Third is distribution analysis. How is this thing spreading? And fourth, temporal progression. Basically, when did it start? You've got to pay really close attention to that timeline. A super sudden onset almost always points to a non-living environmental event or maybe a chemical burn, whereas a living pathogenic spread is going to be way more gradual.
Let's move on to Section 2, reading the leaf patterns, looking at macro to micro clues.
It is so incredibly interesting how the distribution of damage tells us a really vivid story. Check out how a uniform distribution of damage, where literally an entire row or all the plants in a low-lying area are affected, usually means a soil or abiotic issue. Things like extreme soil pH or even a lightning strike, they hit multiple plants all at once. But on the flip side, a random scattered pattern, that points directly to a living airborne pest or pathogen that's just moving unpredictably through the air, jumping from plant to plant.
Now, when you're examining an individual leaf, look really closely at the veins. Seriously, this is a dead giveaway. Fungal leaf spots tend to cross the veins recklessly, just expanding outward in these big circular shapes. But bacterial spots, they actually get physically stopped by the plant's veins. And because of that, they create these very distinct, harsh, angular shapes or streaks right in the leaf tissue.
Next up, Section 3, deciphering terms. Let's break down some diagnostic terminology.
So, a super crucial part of this is understanding terms like chlorosis. Chlorosis is just a fancy word for the yellowing of plant tissue. It's basically a primary cry for help from your garden. But to solve the case, we have to pinpoint exactly where it's happening on the leaf. For example, if you're seeing intervenal chlorosis, which is where the tissue turns yellow but the veins themselves stay nice and green, you're very often looking at a nutrient deficiency. It's screaming that it lacks iron, zinc, or maybe manganese.
All right, let's train your eye to spot necrosis. This is when dead tissue turns brown or black. It is very often the destructive calling card of a fungal infection or some kind of severe blight. It's basically the physical ruin left behind after a pathogen has just completely ravaged the plant's cells, leaving these dry, leathery, or even powdery dead zones behind.
Which brings us to Section 4, the suspects. Time to identify the culprit. When we compare our main culprits side by side, you start to see some really distinct differences. Fungi are just messy, They leave highly visible signs like spores, fuzzy mycelium, or powdery dust. Bacteria, on the other hand, often create these water-soaked, oily, or kind of slimy, oozing lesions. Gross, but helpful for us. But viruses? Oh, viruses operate entirely under the radar. They leave absolutely no visible spores or ooze, which makes them incredibly sneaky.
Right, so unlike fungi and bacteria, viruses aren't going to leave localized, crusty lesions on the outside of a leaf. Instead, they cause what we call systemic viral disruption. They actually invade the plant cells themselves and cause systemic chaos from the inside out. And because they travel right through the plant's vascular system, you're usually going to see the most pronounced, bizarre symptoms popping up in the plant's newest growth. We're talking severe stunting or totally distorted, crinkled leaves.
And that leads us perfectly into Section 5, our case study, AMV, the Alfalfa Mosaic virus.
Looking at the rap sheet for the Alfalfa Mosaic virus, or AMV, we can see it has a massive geographical footprint. I mean, it is found worldwide, and its host range is equally staggering. It hits chili, soybeans, legumes, alfalfa, tomatoes, you name it. When it strikes, it severely stunts the plants and results in little to absolutely no fruit production. It's just a total yield killer for farmers and home gardeners alike.
Here's your hallmark visual clue right here. Take a look at these soybean leaves displaying that classic AMV mosaic pattern. Notice that weird, irregular, mottled mix of bright yellow and dark green tissue? It completely disrupts the normal uniform leaf color. This is not just a simple nutrient deficiency, guys. This is the virus literally hijacking the plant's internal cellular machinery and totally destroying its ability to photosynthesize properly.
Checking out another angle of our crime scene, this chili plant really proves how the virus alters leaf tissue systemically. Look really closely at it. There are no outward physical spores, no fungal fuzz, no gross bacterial ooze. There's just severe yellowing, stunting, and twisted leaves all throughout the entire plant structure. It is the perfect undercover operation.
But here's a highly critical fact. Viruses literally cannot travel alone. They don't have legs, they don't blow in on the wind like fungal spores do. No, they rely entirely on accomplices. We call these physical vectors. A vector is absolutely necessary to pierce the plant tissue and introduce the virus into a tiny wound in a new healthy host.
So, meet the primary vector and main accomplice for the Alfalfa Mosaic virus, the aphid. These tiny little sap-sucking insects essentially act like dirty syringes. As they feed on an infected plant, they pick up the virus. Then, when they mosey on over to a healthy plant to feed again, bam, they transmit the disease directly into the new plant's vascular system.
And that brings us to the pretty harsh reality of this specific diagnosis, which is perfectly captured by this quote right from our sources. It says, "Viral infections cannot be cured. The entire plant must usually be removed to prevent spread." Yeah, it's brutal. Once the Alfalfa Mosaic virus is inside that plant, there is no chemical spray, no magic cure. Complete removal of the infected plant, along with managing the weeds that harbor those aphid pests in the first place, are your absolute only options to save the rest of your crop.
So I'm going to leave you with this final kind of provocative thought. If plant viruses are systemic and completely incurable, is your best defense entirely about prevention? Knowing you can't cure a virus like AMV once it's there, how are you going to use that disease triangle? Managing your host plants, controlling the insect vectors, and altering the environment to protect your garden before the culprit even arrives.